In this video, the basics of enzymes, the learning objectives are to define a biological catalyst, to describe the nature of interactions between enzymes and their substrates, to describe the characteristics of an enzyme's active site, and to understand the lists of do's and don'ts of enzymes. These are a list of features of enzymes that are common misunderstandings. So most enzymes are proteins, and enzymes are biological catalysts. So these are proteins like beta amylase here, and you can see it's a protein. It's got these, right? These are alpha helices. Here's some beta pleated sheets over here. There's beta turns connecting the alpha helices to one another. So this is a protein called beta amylase, and it is an enzyme. It is a biological catalyst. It speeds up biochemical reactions without being consumed in the process. Furthermore, the name amylase gives you a clue that it's an enzyme. Enzymes, the names of enzymes tend to end in ase. Protease, DNase, amylase, kinase. Now that doesn't mean every single enzyme ends in ase, but most of them do. On top of that, the name gives you a clue of what they might catalyze. So, for example, a protease is an enzyme that catalyzes the hydrolysis of proteins. Amylase is an enzyme that catalyzes the hydrolysis of amylose starch. Substrates are the name for the given to the molecule that binds to an enzyme. The molecule that's going to be catalyzed, that's going to be involved in that reaction. And finally, got a little asterisk up here near most because not all enzymes are proteins. Some enzymes are RNA molecules. So for example, ribosomes are made out of RNA and protein, but it's the RNA component of ribosomes that catalyzes the production of peptide bonds between amino acids. Enzymes have very specific substrates, and again, substrates are the name given to the molecule that binds an enzyme. So here, all these red boxes and these hexagons and these H's and minuses, this is all part of a protein. This cleft right here that's holding the substrate, that's called the active site. And it is the active site of the protein that interacts with the substrate, and they do so via weak interactions. Hydrogen bonds, hydrophobic interactions, ionic interactions. Some enzymes bind just one single type of substrate. Some enzymes bind several different types of substrates. But in general, enzymes are very specific for the things that they bind. If we take a little bit more closely, if we look more closely at the active site of an enzyme, we'll notice a few things. Number one, it's this cleft or this crevice here to which the substrate fits in. It is only a small part of the protein. So here is the active site, but look at all of this other part of the protein. And some of that is structural. It's just simply there to hold the active site, but some of them can be very functional. Much of this protein might be subject to regulation, to various other molecules that bind it and regulate that enzyme. We'll talk a lot more about that later. If we look more closely at that active site and compare one active site to another, we'll see some commonalities, that they have this unique microenvironments of just the right pH, just the right ionic strength, and that we see that the substrate is going to bind, like we saw in that previous slide, is going to bind to that active site via many weak interactions. You're going to learn all of this in subsequent videos, but I want to just bring it all here right now. If we have a reaction, a reversible biochemical reaction between A and B, and then it's catalyzed by an enzyme, there are certain things that enzymes can do and certain things that they don't or can't do. So here we have this hypothetical reaction. Here it has a delta G of positive 20 kilojoules per mole. And the absolute first thing that you have to know is that if the delta G is positive, what does that mean about this reaction? Does it mean that it goes in the forward direction? Does it mean that it goes in the reverse direction? Does it mean that it just doesn't go at all? Well, we know that it does more than not go at all, because not going at all means it has a delta G of 0, right? That it is at equilibrium. This has a positive delta G, which means that it's going in the reverse direction. B is get converted to A based on this delta G. So, based on all of this, what can enzymes do? What can they not do? Well, enzymes do speed up biochemical reactions. We knew that. Enzymes do lower the activation of energy of reactants. Well, that's how they speed up reactions, and you'll learn that a little bit later. Enzymes do catalyze both directions of a reaction. Okay, so an enzyme can catalyze the production of B from A or A from B. Now, which direction does it catalyze at any given moment? That's dictated by the current delta G. The current delta G is positive 20. Therefore, if you add an enzyme, it's going to catalyze, it's going to speed up the rate at which B is converted to A. Enzymes don't change the delta G of a reaction. Okay, so if the delta G of a reaction is positive 20 and you add a whole bunch of enzyme to it, the delta G is going to stay positive 20. The enzymes don't change the direction of the reaction. So at this particular set of conditions, no matter how much enzyme you add to that reaction, it's going to go in the reverse direction from right to left, because what the delta G is telling us. And finally, enzymes don't affect the equilibrium of reactions. Okay, the delta G of this is positive 20. That this reaction is going to go from B to A until we hit equilibrium. And that equilibrium is going to be the same even if we have enzymes. The only difference is the rate at which the reaction reaches equilibrium. With an enzyme, it's going to be faster. Without an enzyme, it's going to be slower. So again, be sure to review this video. Make sure you can define biological catalyst. Make sure you can describe the nature of interactions between the enzymes and their substrates. Make sure you can describe the characteristics of an enzyme's active site. And finally, and this will become more and more clear as you watch more videos, understand the list of do's and don'ts of or for enzymes.